Hello student, welcome to our channel. Today we'll discuss about zoology subject from cytology chapter. The first question is that the backbone of DNA molecule is made of OKI, the staron, alternative sugars and phosphates. Okay. Next question. According to Chargaff's rules, the amount of adenine is equal to that's thymine T. That means Chargaff states that in any species DNA the amount of A equals to T. That means adenine equals to thymine and the amount of G equal to C. Okay, G means guanine and C means cytosine. Okay, due to specific base of pair. Next question, which of the following is not a DNA nucleotide? कौन सा DNA न्यूक्लियोटाइड नहीं है? वो है राइबोज शुगर. ओके, DNA contains deoxyribose sugar, but ribose sugar is found in RNA nucleotide. Okay. Next question, the two strands of DNA double helix are held together by that's hydrogen bonds. Okay, hydrogen bonds form between the complementary base pair, that means between adenine and thymine, 3 between G and C. Next question, which nitrogenous base is found in RNA but not in DNA? That's uracil. क्या होता है वो? Uracil. In RNA, uracil replaces thymine and pairs with adenine. Synthesizing an RNA molecule from a DNA template is called क्या कहता है? ट्रांसक्रिप्शन कहा जाता है. ट्रांसक्रिप्शन. ओके. नेक्स्ट, synthesize a protein from an mRNA template is called mRNA. That's translation. क्या कहता है वो? Translation. Next, which RNA carries the genetic code from the nucleus to the ribosome? That's mRNA. क्या होता है वो? mRNA. Next, which RNA brings amino acid to the ribosome? Which RNA brings amino acid to the ribosome? वो क्या होता है? tRNA. क्या होता है वो? tRNA. Next, a sequence of 3 nucleotides on a mRNA that codes for an amino acid is codon. Next, DNA replication is a semi-conservative because each new molecule has on old strand and on new strand. Okay. Next, the enzyme that unwinds the DNA double helix is helicase. क्या होता है वो? Helicase. Next, the enzyme that adds new nucleotides during DNA replication. क्या होता है वो? DNA polymerase. DNA polymerase. Next, short fragments of DNA synthesized on the lagging strand are called Okazaki fragments. Okazaki fragments. Next, the enzyme that joins Okazaki fragments together is DNA ligase. क्या होते हैं वो? DNA ligase. That means the DNA ligase forms the final phosphodiester bond to seal the nicks between Okazaki fragments, creating a continuous DNA strand. Next, which is the primary function of rRNA? आरएनए का प्राइमरी फंक्शन क्या होता है? कंपोनेंट ऑफ राइबोसोम. नेक्स्ट, in eukaryotes, non-coding sequence removed from pre-mRNA, thus introns. Next, the structure of DNA has the structure of DNA was first described by Watson and Crick in 1953. Okay, that means James Watson and Francis Crick, using X-ray diffraction data from Rosalind Franklin, proposed the double helix model for DNA in 1953. Next, the two strands of DNA molecule are antiparallel. This means they run in opposite 5 to 3 direction. Okay. Next, the central dogma of molecular biology is that is RNA, DNA, RNA, protein. DNA, RNA, protein. Next, the start codon for translation is OG. Okay. Next, in DNA purine bases are that is adenine and guanine. The complex of DNA and protein in eukaryotic chromosomes is called chromatin. क्या होता है वो? Chromatin. What is the role of primase in DNA replication? Synthesize a short RNA primer. Next, the phenomenon where a single gene can code for multiple proteins. क्या होता है वो? Alternative specializing. Next, which of the following is a stop codon? Stop codon क्या होता है? UGA. UGA. Next, the ends of eukaryotic chromosomes are protected by telomeres. क्या होता है वो? Telomeres. Which enzyme is responsible for transcription? RNA polymerase. क्या होता है वो? RNA polymerase. The oval hypothesis relates to the pairing between the third base of a codon and anticodon. Codon and anticodon. A mutation that changes a codon for an amino acid into a stop codon is a nonsense mutation. Next, the basics repeating unit of chromatid is the क्या होता है वो? न्यूक्लियोसोम होता है. Next, which bond connects nucleotides in single DNA strand? वो phosphodiester bond. Next, reserve transcriptase is an enzyme that synthesizes DNA from RNA. That means found in retroviruses like HIV. Reverse transcriptase violates the central dogma by creating DNA from an RNA template. Next, adding a cap and tail to eukaryotic pre-mRNA is known as RNA processing. The genetic code is degenerated. This means most amino acids have more than one codon. Next question: the prokaryotic genetic material is typically single and circular chromosomes. Okay. Next, the A, P, and E sites are found on the large ribosomal subunit. Next, the enzyme that relieves supercoiling stress ahead of the replication fork is topoisomerase. Next, the process by which bacteria take up foreign DNA from the environment is called transformation. क्या होता है वो? Transformation. Next, the frame shift mutation is caused by वो deletion or insertion of non-3 bases. Okay. Next, the 3 poly A tail on a eukaryotic mRNA is increased mRNA stability. The Hershey-Chase experiment used radioactive phosphorus to level up what? That's DNA. क्या होता है वो? DNA. Okay. Next, which of the following is a post-translation modification? That's glycosylation. Okay. Next, the enzyme telomerase is important for replicating the ends of linear chromosomes. Next, which organelles is surrounded by a single membrane? वो क्या होता है? Lysosome. That means mitochondria, nuclei, and chloroplasts are double membrane, but lysosomes, peroxisomes, and vacuoles are enclosed by a single membrane. Next, what is the role of the signal recognition particle, that's SRP, that is direct ribosomes to the ER. ER मतलब endoplasmic reticulum. Okay. Next, the AIMS test is used to identify potential the mutagens. Next, apoptosis is also called as programmed cell death. Okay. Next one, mitosis one is called reductional division because it separates homologous chromosomes. Okay. Next, mitosis and meiosis 2 are called equational division because they are separate sister chromatid. Okay. Next, which is the correct hierarchy of genetic material organizations? That's nucleotide, gene, chromosomes, and genome. Next, the process of charging a tRNA molecule involves attaching it to that means what? The specific amino acid. The histone octamer is composed of two copies each of which histone protein. Then H2A, H2B, H3, H4. Next, the TRP operon is a traceable operand because its transcription is inhibited when tryptophan levels are high. Okay. Next, in DNA gel electrophoresis, DNA fragments migrate towards the क्या का वो होता है towards the positive and electrode because they are in negative charge. Next, the lac operand in E. coli is an example of an inducible operand. क्या होता है inducible operand. Next, the bar body is an example of an inactivated X chromosome. What is the function of capsepsis? That's क्या होता है वो key enzymes in apoptosis. The arrangement of microtubules in a central is 9 triplets in a ring. Which organelle is a major site for calcium ion storage in muscle cells? In muscles, so that is sarcoplasmic reticulum. Okay. The missense mutation is a point mutation that changes on amino acid to the another. Next one, ribosomes are क्या होता है राइबोसोम? RNA molecules with catalytic activities. Next one, leptotin, zygotin and pachotin, diplotin and dikinesis are sub-stage of prophase 1, that is meiosis. The term karyokinesis refers to division of nucleus. Okay. Then, if a cell has a diploid number of 46, how many chromatids are in prophase 1? 92. क्या कितना होता है वो? 92. That means in prophase 1, each of 46 chromosomes has already replicated. So each consists of 2 sister chromatids. Therefore, the total number of chromatids is 46 into 90, 46 into 2. That means 92. The tumor suppressor protein p53 is known as the guardian of the genome. Okay. Next, the cell function allowing communication between adjacent animal cell is the gap junction. क्या होता है gap junction. In plant cell, similar communication channels are called plasmadomostata. It's the last questions in this video. So subscribe our channel for more video, and we'll do every session video what you need for your competitive exams. Thank you.